Starting with A.4.1, we have that molecular phylogeny is the process by which you determine how closely two species are related to one another at a molecular level. We also have that nucleotides and amino acids are the same in all organisms, and this is universal, indicating that there is a common origin of all life. Also have in mind that mutations are random, but over millions and millions of years, the average is actually steady. And this means that when these two monkeys split or diverged, each lineage began to accumulate its own random mutations, and this is going to lead to speciation, which we're going to look at later in the video. So this is a hemoglobin beta chain comparing different species and their amino acid similarities and differences with humans. So we'll see that monkeys have the most similarities and the least differences. Then it's dogs, birds, and fish. Selective breeding is the choosing of the individuals of a species with desirable characteristics. In the case of dogs, it would be the friendly dogs, the more tameable dogs, and making them reproduce to pass on those traits, giving us the breeds that we have today. We also have that farmers will selectively breed their cows to increase milk production and their crops to increase disease resistance and the amount of yield. A great example of this is the wild mustard plant that gives us broccoli, cabbage, cauliflower, and kale. So imagine how useful this plant is. We can link this to Darwin's theory of evolution, where he stated that humans cause the selective breeding and nature causes natural selection when studying pigeons. And he stated that selective breeding is faster as humans deliberately pick the traits. If we look at the ancestors of giraffes, we'll see that there was a variation in their neck length. So some had longer necks, some had shorter necks, and this did not really matter because the tree branches of this species were at lower heights anyway. Now, environmental changes would lead to these tree branches growing taller and hence leading to food shortage, and only the giraffes with the longer necks would have an advantage and would actually survive, passing on their genes to descendants. And this is an example of natural selection, a big part of Darwinian evolution, or Charles Darwin's theory of evolution. And lastly, because those with longer necks survived, now longer necks became more common as they passed on their traits and desirable characteristics. Now looking at Lamarckian evolution and using the same example of giraffes, we'll have that there was an initial variation in neck length, which did not impact their survival, and the same environmental change of taller tree branches. This time it stated that because the giraffes stretched their necks to reach the taller tree branches, this led to the elongation of such, and this was passed on to offspring as a genetic trait. Now comparing Lamarckian and Darwinian evolution. So starting with Darwinian evolution, we have many different points. We haven't looked at overproduction, so essentially it's when there's too many offspring for the environment to support, leading to competition and survival of the fittest, which very much links with natural selection. Then we have the inheritance of the favorable traits and the adaptation and speciation. So this involves reproductive isolation. Don't worry about these two keywords. We're going to look at them later in the video. Then Lamarckian evolution is when there's the increased use or disuse of a certain organ, and this leads to the change in size of such organ, and hence the passing on of this trait onto offspring. Homologous structures have the same structural plan but different functions. So looking at these vertebrate limbs, we'll see that they each have 5 digits, as you can see here, 3, 4, 5. And this is a sign of divergent evolution, which is essentially when ancestral species adapt to different environmental conditions according to the pressures of natural selection. So we'll see that bats and humans each have 5 digits, but divergent evolution caused them to have different functions. So the human hand is used for its respective functions, and the bat's wing is used for flying. Okay, so this is an example of divergent evolution. The opposite would be analogous structures, which have the same or similar functions but different structures. So the main example of this would be the bird versus the insect's wing, and this is convergent evolution, where distantly related groups of organisms face similar environmental conditions and hence adapt in similar ways. A species can be defined as a group of organisms that can mate with each other, producing fertile offspring and sharing a common gene pool. Speciation is the process by which new species are formed by the modification of the original gene pools of a species into separate gene pools in a way that interbreeding is prevented. That's going to make sense in a second with reproductive isolation. So this involves the barriers that prevent populations of the same species from interbreeding. They can be physical, such as rivers, mountains, etc. They can also be distance barriers, which involve the organisms of the same species being too far away from each other in order to reproduce. Or they can also be human barriers, such as roads, buildings, etc. And now looking at reproductive isolation. Looking at the 4 steps of reproductive isolation, to the left you'll have the written step, and to the right you'll have the nice little diagram that I've prepared. So firstly, we'll have that a population occupies a habitat. So far so good. Step 2, a physical barrier such as a river splits it into 2 evolutionary forces that act independently. Step 3, generations later, populations A and B are now genetically different due to experiencing different environmental conditions and random mutations accumulated over the millions of years. And lastly, step 4, they are now different species because they cannot interbreed anymore. So this is speciation caused by reproductive isolation. Also have in mind this formula for selection differential, because you could possibly get a maths equation on this. You wouldn't want to miss out on the formula, would you? Moving on to A.4.2 biodiversity. If this video is helping you so far, make sure you tell me in the comments. If it hasn't, you should also tell me so I improve. And yeah, just make sure to leave a like as well. Starting with genetic diversity, we have that it's the variation in genes within a population, often caused by mutations and differences in alleles. But the important thing here is that it is essential for survival, because when there's more variation in genes, it's more likely that a certain individual in the species will have the beneficial trait required for survival at that time. So those with less genetic diversity have more of an extinction risk. An example of great genetic diversity that led to survival is the DDT insecticide mosquito, which developed resistance to this insecticide because some individuals in the species already had resistance to this insecticide, and hence they survived and passed on those traits onto offspring. So this is another example of natural selection. Now, looking at species diversity, we have 2 definitions. So there's the richness, or the amount of different species in the area, and the evenness, so how equal the populations are with one another. Lastly, we have ecosystem diversity, which is, as the name suggests, the variety of ecosystems, both terrestrial and aquatic, found in a given area. So rainforests have high ecosystem diversity because they have both the aquatic and the terrestrial habitat. The current mass extinction happening in the world right now is anthropogenic, which essentially means human caused. So this can be overexploitation, or the excess of certain activities such as hunting, harvesting, or fishing, the habitat loss from deforestation and other unfortunate situations, the climate change, which is natural but caused by humans, the pollution, and the introduction of invasive or alien species to habitats where they don't belong. So for example, humans will import certain animals or organisms from Africa to America or to Asia, which are invasive or alien species because they are not from the local area and they can alter the ecosystem there. Looking at 2 causes of ecosystem loss, we have dipterocarp forest, which are very important because they store large amounts of carbon, which mitigates climate change, and the trees have roots that prevent soil erosion. Problem is there's a lot of logging and palm oil plantations going on, and this increases the loss of this ecosystem. Same can be said with coral reefs, which are made of the coral polyps, the animals, plus their exoskeletons, and the symbiotic algae that give them the color. So these coral reefs are sources of medicine, and they protect coastlines from erosions and storm. So these are their benefits. The problem is certain things such as pollution, overfishing, and climate change, which are anthropogenic, so human caused, are mitigating these benefits. The available evidence for this biodiversity crisis starts with the ICUN's list of species and ecosystems that are currently threatened with extinction. So we can see that there are 42,000 of them around here. Then we have the citizen science data. So essentially there have been declines in certain populations of species over harvesting pressures. An example of this would be the amount of horseshoe crabs at the moment. Then we have the IBPES, which states that over 1 million species are endangered at the moment. And lastly, we have that certain biodiversity surveys have shown that species evenness and richness has declined over the years. Now, let's look at the causes of biodiversity loss and the conservation methods to play against this. Habitat fragmentation would be a cause of biodiversity loss, as well as habitat destruction, the introduction of invasive or alien species, the spreading of pests and diseases caused by human traveling, the pollution such as sulfur dioxide, which turns into acid rains, and this is increased by human activity as well, and the overexploitation that we saw before. As a result, the current extinction rate is 100 to 1,000 times higher than it would be in natural conditions. Multi-pronged conservation is the method used at the moment because different species require different measures, and this leads to in situ conservation, so protecting species in their natural habitats, such as in national parks, and the opposite, which is ex situ, where we protect these species in our own terms, in captivity. So looking at examples for each, we'll have that captive breeding right here is, as I said before, keeping them in our own terms, such as in zoos, breeding centers, etc. We also have gene banks and sperm banks, though we're not keeping the animal itself, but keeping their genes and sperm is also very important. So the goal of this is to one day reintroduce them into the wild. However, the drawbacks involve the low genetic diversity that we know is bad for survival, as we saw from before. Then we have the rewilding part in situ conservation. So when we fix the problems we've caused, so we'll reintroduce the missing species and rebuild their ecosystem. Now, restoring their land and water damaged by human activity, such as deforestation, is what we call keyword reclamation of degraded ecosystems. A3.1 variation. Please give me tips on how to improve my videos and leave a like. Thank you. As a response to the variation between species, we have developed what's called the morphological species concept, where they are named differently based on their physical traits. So the big cats are going to be the Panthera, and we have that the tiger is Tigris and the lion is Leo, using the language Latin. Then we have binomial nomenclature, where this time everything will be in italics, and the first word is going to be the genus, which is going to have an uppercase. So we'll see here, there's a capital P at the start of Panthera, and then the second word is the actual species that starts with a lowercase. Now let's have a look at the biological species concept. The main point of this concept is that different species cannot produce fertile offsprings, and that for fertile offspring to be produced, it has to be done by the same species, okay? So an example that supports this is when two different species, the donkey and the horse, reproduce, they'll produce what's called a mule, which is infertile. Okay, so this supports this concept. To have fertile offspring, they need to be the same species. Now, an example that plays against this is when the tiger and the lion reproduce. Yes, because this can give birth to a liger, which is actually fertile. Okay, so two different species have produced fertile offspring. And this again objects the concept. In eukaryotic cells, our chromosomes are located in the nucleus of our cells. So remember that eukaryotes are plants, animals, and fungi. Now, diploid cells will always have an even number of these chromosomes, and sudden chromosome changes that can happen in situations such as errors in cell division can cause speciation that we've seen before. Now, in humans, we'll have 22 pairs of autosomes plus 2 sex chromosomes, which gives us a total of 46. And then we have chimpanzees that have 23 pairs of autosomes and 2 sex chromosomes as well, giving a total of 48. What does this mean? This means that human chromosomes were formed by the fusion of 2 ape chromosomes. This is going to make sense now when we look at karyograms and karyotyping. Karyotyping is the process by which we isolate certain chromosomes and using size, banding patterns, and centromere location, as you see here, we arrange and pair them. So we'll see that there are different sizes around here. You'll see that this one's smaller than 21 is smaller than 1, and so forth. And we have the centromere location. So the centromere in this chromosome would be about here, whereas in this chromosome it would be about here. Okay, so we use these little factors to arrange them. Now the sex chromosomes are XX for females and XY for males, as you can see right here, where X is longer and hence contains more genes than Y. Okay, so the karyotyping is the process by which we isolate and stain the chromosomes, and the karyogram is this that you can see right here. Okay, so think diagram, karyogram. Now, looking at another karyogram, we have that if we're comparing human chromosomes and chimpanzee chromosomes, the genes, as you can see here at 2, match those of the 2 ape chromosomes. Now, this is a bit complicated to understand. It's more of just memorizing it. But there's telomeric DNA found at the fusion site. Just remember that. And second, non-functional centromeres are present as well. A genome is all the genetic information that an organism contains, and it can be changed by single nucleotide polymorphisms. So here we'll see that these three people have the same base sequences or nucleotides around here, as you can see, but have different ones when they are encircled by the single nucleotide polymorphism. So there's A, G, and T. Now this is enough to increase genetic diversity, and this often happens in non-coding regions of DNA. Also have in mind that genomes differ in size and base sequences, okay? And we're going to see that in this table where the hippopotamus has the most genome size, having 2,424.26 million base pairs. Another thing to have in mind is genome sequencing, which is the process of determining the complete DNA sequences of an organism's genome. Okay, so this is how we figure out how big its genome actually is. And the functions of this process are to diagnose diseases, track evolution, form personalized medicine, and predict risk of future diseases before they actually develop. So it's a very useful technique.